Dear students, welcome to the massive open online course. Today's topic is History of Human Genetics. The main objectives of this module are to know about the origins of human genetics, to know about the historical perspective of how genetics has evolved as a science, to know about contributions of various scientists in human genetics, to understand the clinical relevance of genetic concepts, to understand the scope of genetics. The history of human genetics is very interesting since as the concept of human genetics have often influenced social and political events. At the same time, the development of human genetics as a science has also been influenced by various political forces across the globe. Since the dawn of civilization, man used the concept of heredity for the improvement of cultivation of crops and domestication of animals. For example, pedigrees of horses with inherited traits and cross-pollination of palm trees. But the first documented speculation on heredity have been reported at the time of ancient Greeks, and some of their ideas are still considered relevant today. Already in ancient times, first considerations and thought on the inherited difference of man were made. Plato wrote in his state Utopias how carefully a partner for producing children should be selected. The history of genetics dates from the classical era with contributions by Pythagoras, Hippocrates, Aristotle, Epicurus, and others. The most influential early theories of heredity were that of Hippocrates and Aristotle. In 400 BC, Hippocrates, as the father of the medicine, believed in the inheritance of acquired characteristics. According to these theories, during sexual intercourse, the organs of the body of a parent transmitted as an invisible seed which looks like a miniature-sized baby in the mother's womb. Later in 300 BC, Aristotle proposed the role of blood in heredity, wherein semen and the menstrual blood are the purified blood. The hereditary essences make the baby. Even today, people still speak of certain traits as being in the blood and of bloodlines and blood ties. Then for a long time, the question of heredity was no longer of interest until in 1605 when Louis Mercado published De Morbis Hereditas. This publication was followed by similar ones in the following years. More than 200 years later, Joseph Adams published his influential book, A Treatise on the Supposed Hereditary Properties of Diseases based on clinical observations. But the question, how does heredity work, was still unsolved till 1814. In the middle of the 19th century, two important publications were made. One was the Francis Galton, who published Hereditary Character and Talent. The second one was the Modern Genetics, began with the work of Gregor John Mendel, his work on pea plants, published in 1866, which established the theory of Mendelian inheritance. Gregor John Mendel's idea was different from that, and he talked about influential hereditary factors called now as genes, which determine the distinct differences between individuals. These genes are transmitted through sperm and egg and guide the development of the offspring. Therefore, the genes are responsible for reproducing the distinct features of both parents that are visible in their children. However, most of the mechanisms of heredity remained a mystery until the 19th century when genetics as a systematic science began. Gregor John Mendel, experimenting with peas, announced his attempts with plant hybrids in 1866, like Versuche über Pflanzen Hybride, and set up the paradigm of Mendelism. But it was not until the 20th century that William Bateson in 1906 he rediscovered the Mendelism and named it as genetics and influenced the British medical societies. In 1888, Wilhelm G. von Waldeyer Hartz introduced the term chromosome. At the beginning of the 20th century, many articles were published about these, though the human chromosome number was established late in 1956. Gene mapping was important for the development and practice of human genetics. The first diseases were classified as hereditary early, for example, polydactylia and hemophilia. The discovery of the inheritance of alkaptonuria by Archibald E. Garrard in conjunction with William Bateson was a path-breaking development. The year 1900 marked the rediscovery of Mendel by Hugo de Vries, Karl Correns, and Erich von Teschermark, and the basic principles of Mendelian genetics had been studied in a wide variety of organisms. Most notably among them are the fruit fly Drosophila melanogaster, led by the great scientist or genetist Thomas Morgan and his fellow Drosophilists, who developed the Mendelian model, which was widely accepted by 1925. Alongside experimental work, mathematicians developed the statistical framework of population genetics, bringing genetic explanations into the study of evolution. With the basic patterns of genetic inheritance established, many biologists turned to investigations of the physical nature of the gene. In the 1940s and early 1950s, experiments pointed to DNA as the portion of chromosome and perhaps other nucleoproteins that held genes. A focus on new model organisms such as viruses and bacteria, along with the discovery of the double helical structure of DNA in 1953 by James Watson and Francis Crick, marked the transition to the era of molecular genetics. The description of genetic implication of the structure of DNA, that is deoxyribonucleic acid, by Francis Crick and James Watson, was the starting point for the genome concept. This began to influence human genetics as well as molecular biology and raised the questions of its influence, and at least uncertainty gained importance in diagnosis. As biochemical and chromosomal diagnosis of genetic disorders progressed, the need for counseling for families and patients was soon recognized. The first center, Diet Clinic, was founded in 1941 at the University of Minnesota, USA. This was the starting point, and by 1955, there were already 20 institutions in the USA that offered counseling and information free of charge to the general public. After the heredity of characteristics and qualities was recognized, the first attempts were made to diagnose dispositions and diseases as genetic. This was done using pedigree and family histories, but the results could lead to genetic discrimination of patients. And in the following years, chemists developed techniques for sequencing both nucleic acids and proteins, while many others worked out the relationship between these two forms of biological molecules and discovered the genetic code. The regulation of gene expression became a central issue in the 1960s, and by the 1970s, gene expression could be controlled and manipulated through genetic engineering. In the last decade of the 20th century, many biologists focused on large-scale genetic projects such as sequencing entire human genomes. The word genetics was introduced by English biologist William Bateson in 1901, who propagated Mendel's work and the champion of Mendel's principles of inheritance. Genetics, the study of genes, including the ways they act in cell and the ways they are transmitted from parents to the offsprings. Hence, in this module, we will discuss the following aspects now. The first one is origin of genetics. The source of genetics lies during the time of development of theories of evolution by Wallace and Charles Darwin in 1858, who developed the theory of the origin of species by natural selection. However, they did not know that genes had a role to play in this phenomena. Later on, in 1865, Gregor Mendel conducted experiments on inheritance and genetics of pea plants and described the unit of heredity as particle without change passed on to offsprings. He observed that organisms inherit traits or characters by way of discrete units of inheritance, which he called as the traits, but he was not knowing about the genes. He provided a mathematical foundation of the science of genetics, but the scientific community did not recognize his work at that time. This work is the basis of understanding the principles of genetics even today. For this contribution, Gregor John Mendel is known as the father of genetics. During his time, most biologists had the idea that characteristics passed on to the next generation through blending of inheritance and traits from each parent averaged. Later on, Ernest Haeckel in 1866 correctly predicted that the hereditary material is located in the nucleus. Friedrich Miescher in 1869 found both phosphorus and nitrogen in the nuclei of white blood cells in pus. The material first named nuclein, as it seemed to come from the cell nuclei, was later called as nucleic acid after 1874. But with this, he was not able to conclude the role of nucleic acid in inheritance, since a single DNA molecule alone could not provide the level of variation observed in nature within and between various species. Mendel's work was rediscovered in 1900 by three botanists, Hugo de Vries of Holland, Karl Correns of Germany, and Erich von Teschermark of Austria, and with this, after a hibernation period of 35 years, the science of genetics was finally born. In 1902, William Bateson published an English translation of Mendel's German text, that is Mendelism, the Principles of Dominance, Segregation, and Independent Assortments, which we know by the name of Three Laws of Mendel. In 1902, Walter Sutton and Theodore Boveri led the development of chromosomal theory of inheritance and established the hereditary material resides in chromosomes. In 1908, Archibald Garrod proposed that inborn errors of metabolism caused human disease, alkaptonuria, and certain other hereditary diseases. During the period 1905 to 1930, extensions of Mendelian principles of genetics with linkage, Hardy-Weinberg principle of genetic equilibrium, genetic mapping, number of linkage groups, number of chromosomes, sex linkage, lethal genes, and maternal inheritance were established. This study led to the understanding of the mechanism of transmission and function of genes. Now, let us focus on the period of human cytogenetics. Cytogenetics is the study of chromosomes. their inheritance and functions. Virchow published the first diagram of mammalian chromosomes in 1865 from the testes of a cat. Walter Fleming in 1879 developed a new staining technique to identify chromosomes, which allowed observation of the chromosome behavior in the cell nucleus during normal cell division. In 1882, he coined the term mitosis for cell division. Von Beneden discovered the number of chromosomes in any body cell is constant, and the number characterizes a particular species. In 1887, he also contributed to the discovery and naming of meiosis. In the same year, Weizmann predicted the reduction division in the form of meiosis. Heinrich von Waldeyer coined the term chromosome to describe them in the year 1888. In 1901, McClung named the sex chromosome, which was different in males and female grasshoppers. Sutton and Bowery in 1903 independently hypothesized that chromosomes are the hereditary units. Later in 1910, Thomas Hunt Morgan showed that genes reside on chromosomes while determining the sex-linked traits in Drosophila. In 1911, one of Morgan's students, Alfred Struant, invented the procedure of linkage mapping in Drosophila and made the first genetic map of a chromosome in 1913. The progress of human genetics is as follows. The genetics before 1952, that is the Dark Ages. It took many years to settle the most fundamental question in cytogenetics, that is, what is the chromosome number in a normal diploid human cell? Winwater in 1912 reported 47 chromosomes in sperm and 48 in egg, concluding an XX and X0 sex determination mechanism. Later on, Painter in 1922 revised his opinion to 48 from 46 and correctly insisted that humans have an XX-XY system of sex determination. Painter's study on human chromosomes was by arresting cells during division and visualized them. He also used staining techniques which allowed for limited differentiation of specific chromosomes based on darkly versus lightly stained areas. The number of human chromosomes remained 48 for over 30 years in science books. Now, let us discuss the second part, that is genetics between 1952 to 59, that is hypotonic period. During this period, an American cytogenetist, Husu, used a solution with a lower salt concentration, and the cells absorbed water through their membranes and swell, but not burst. However, chromosomes readily separated and making them easier to count. In 1956, Tijo and Livon had analyzed 265 preparations from 22 different cell cultures made from the embryonic lung tissues of 4 different legally aborted embryos. They reported that the karyotype of females was 46 XX and the males with 46 XY. Now let us discuss the third, that is genetics between 1959 to 1969, that is trisomy period. The development of the method to determine the human karyotype enhanced discoveries of a variety of aberrations in the number and morphology of human chromosomes. The first trisomy 21 was discovered in 1959 by Lejeune and his colleagues. Their paper is still recognized as one of the classical in human genetics. Later on, Klinefelter syndrome by Jacobs and Strong in 1959 and Turner syndrome by Ford and his colleagues in 1959 and Patau's syndrome by Patau and his colleagues in 1963 were discovered. The metaphase chromosomes were then classified into 7 groups based on the Denver classification in 1960s, with revisions at the London conference in 1963 and the Chicago conference in 1966. Now let us discuss the fourth, that is the genetics between 1969 and 79 that we called as the bending era. During this period, 2 techniques revolutionized the study of human chromosomes. The first was a report of Kasperson and his associates in 1970s that certain fluorescent derivatives of quinacrine bound differently to different parts of chromosomes and called as Q-bending. This technique was less than optimal for routine studies as the fluorescent staining quickly quenched. Hence, several other bending techniques were developed, for example, G bending, R bending, C bending, and NOR bending, each having its specific properties and applications. Later on, G bending became the most prominent and frequently used method as it gave better resolution than the Q bending and also allowed permanent preparations and did not necessitate the use of fluorescence microscopy. Prague and Gall in 1970 first reported C bands when they discovered that the centromeric regions of mouse chromosomes are rich in repetitive DNA sequences. Nucleolar organizing region, that is NOR, bending technique stains the NORs of the chromosome. Goodpasture and his colleagues in 1975 developed this simple silver staining technique, which is useful in clinical practice to study specific chromosome polymorphisms such as second satellite. The other technical developments was the high-resolution bending techniques. These techniques help in identifying the small chromosomal aberrations more elegantly. Now the fifth one, that is the genetics period from 1980 onwards. That is also called as the molecular era of genetics. In the 1980s, Purdue and Gall developed a method to visualize chromosomes using fluorescent labeled probes called fluorescent in situ hybridization or FISH in tissue samples. This study allowed the detection of chromosomal abnormalities that involve small segments of DNA. Based on these principles, advanced techniques were later on made, such as spectral karyotyping or SKY and multiplex FISH, that is MFISH, Sky technique as developed by Schrock and his colleagues, in which he allowed the investigator to unambiguously display and identify all the 24 chromosome pairs, that is 22 autosomes and XY, without prior knowledge or any abnormalities involved. There are many ways to attach molecular labels to specific genes and chromosomes, as well as to particular RNAs and proteins that make these molecules easily discernible from other components of the cell, thereby greatly facilitating cytogenetics research. Now let us discuss the period of molecular genetics. Molecular genetics began only after 1941 when American genetist George Beadle and American biochemist Edward Totem showed that the genes are coding for catalytic proteins called enzymes. Subsequent studies in other organisms showed that genes generally code for proteins. Oswald Avery, Colin M. McLeod, and McLean McCarthy showed that bacterial genes are made of DNA. Later, these experiments have studied in all organisms. A groundbreaking discovery was attained in 1953 when James D. Watson and Francis Crick and Maurice Wilkins discovered a double helix model for DNA structure, that is deoxyribonucleic acid, that demonstrated that DNA is capable of self-replication by separating both strands of DNA and use them as templates for the synthesis of new DNA molecules. So each strand of DNA has made up of 4 nucleotides. They also proposed that a specific nucleotide sequence of DNA could contain a code for an amino acid sequence and hence protein structure. Later on, Seymour Benzer in 1955 showed that the mutant sites within a gene and genes are a linear structure. Matthew Meselson and Franklin Stahl in 1958 for the first time experimentally demonstrated the semi-conservative model of DNA replication. Francis Crick and Sidney Brenner in 1961 showed that the genetic code is a triplet code. That is, each codon consists of 3 letters. The Marshall Nirenberg and Hargobind Khurana in 1966 deduced the genetic code of all 64 possible triplet codons and the amino acids that code for. Subsequently, studies in many organisms showed that the double-stranded DNA structure, replication mode, and the genetic code are the same. In this period, various advanced technologies, including molecular biology, recombinant DNA technology, and biotechnology methods, were made much progress in understanding the concept of molecular biology and molecular genetics. Now let us discuss some of the great scientists and their discoveries related to the human genetics throughout the course of the evolution of human genetics. Let us first discuss the contributions made by the Gregor John Mendel, who is also called as the father of genetics for his discovery of Mendelian principles. In 1886, Austrian botanist Gregor Mendel published the results of his experiments with pea plants. His work later provided the mathematical foundation of the science of genetics. Mendel studied the inheritance of traits between generations based on experiments involving the garden pea plant. He deduced that there is a certain tangible essence that is passed on between generations from both the parents. Mendel established the basic principles of inheritance, namely as the principles of dominance, independent assortment, and segregation. Let us now discuss the contributions of Friedrich Miescher. In 1869, Swiss biochemist Friedrich Miescher became the first to isolate nuclein, now known as the DNA. Although he developed a hypothesis explaining the role of nuclein in heredity, he ultimately concluded that one molecule alone could not provide the level of variations observed in nature within and between the species. Let us now discuss the Mendel's law of segregation. In 1900, Mendel's experiments were discovered independently by 3 scientists: the Dutch botanist and genetist Hugo de Vries, German botanist and genetist Karl Heinrich Correns, and Austrian botanist Erich Teschermark von Seyssingegg, giving rise to the modern science of genetics. Let's now discuss the bacterial transformation experiment by Frederick Griffith. Frederick Griffith, English scientist, in 1928 was a bacteriologist, and he conducted experiments suggesting that bacteria are capable of transferring genetic information and that such transformation is heritable into the offsprings. Let's now discuss the contributions of Barbara McClintock. In 1931, American scientists Harriet B. Creighton and Barbara McClintock published a paper demonstrating that new allelic combinations of linked genes are correlated with physical exchange chromosome parts. Their findings suggested that chromosomes form the basis of genetics. Let us now discuss the initial proposal of DNA structure. In 1944, Canadian-born American bacteriologist Oswald Avery and American biologist McLean McCarthy and Colin McLeod reported that the transforming substance, the genetic material of the cell, was DNA. Polynucleotide chain of deoxyribonucleic acid, that is DNA. In 1950, Austrian-born American biochemist Erwin Chargaff discovered that the components of DNA are paired, that is in 1:1 ratio. Thus, the amount of adenine is always equal to the amount of thymine, that is A to T. And similarly, that the amount of guanine is always equal to the amount of cytosine. Let us now discuss the contributions of the Rosalind Franklin. In 1951, British scientists Rosalind Franklin, Maurice Wilkins, and Raymond Gosling conducted X-ray diffraction studies that provided images of the helical structure of DNA fibers. In 1953, using Chargaff's data and the X-ray images recorded by Rosalind Franklin, Maurice Wilkins, and Raymond Gosling, British biophysicists James Watson and Francis Crick determined the molecular structure of double helix DNA, that is the deoxyribonucleic acid. Watson and Crick and Wilkins shared the Nobel Prize for this discovery for 1962 in physiology and medicine. Now let us discuss the recombinant DNA. In 1960s, Swiss microbiologist Werner Arber and American microbiologist Hamilton Othanel Smith and Daniel Nathans discovered restriction enzymes which cleave DNA into fragments, the discovery for which the three men shared the 1978 Nobel Prize for Physiology or Medicine, and this enabled scientists to manipulate genes by removing and inserting DNA sequences, and these restriction enzymes are also known as the molecular scissors in molecular biology. In 1970s, American molecular biologist Alan N. Maxam and Walter Gilbert and English biochemist Frederick Sanger developed some of the first techniques for DNA sequencing. So Gilbert and Sanger shared the 1980 Nobel Prize for Chemistry for their work. Let's now discuss the DNA sequencing development. In 1983, American biochemist Carrie B. Mullis invented the polymerase chain reaction, or the PCR, a simple technique that allows a specific stretch of DNA to be copied billions of times in a few hours. Mullis received the 1993 Nobel Prize for Chemistry for his invention. Let's now discuss the human genetics in the 90s. In 1990, the Human Genome Project, that is the HGP, began. By the time of its completion in 2003, Human Genome Project researchers had successfully determined, stored, and rendered publicly available the sequences of almost all the genetic content of the human genome. Now let us know the International HapMap project. In 2002, the International HapMap project, which was designed to identify genetic variations contributing to human diseases through the development of a haplotype, that is haploid genotype map of the human genome, began. By completion of the phase 2 of this HapMap project in 2007, scientists had a data on some 3.1 million variations in the human genome. Now let us discuss the 1,000 Genomes project. In 2008, the Thousand Genomes Project, an international collaboration in which researchers aimed to sequence the genomes of a large number of people from different ethnic groups worldwide with the intent of creating a catalog of genetic variations, began. The project was completed in 2015. Finally, let's now discuss the scope of human genetics. Genetics is the conceptually complete and most rigorous field of biology. The area of human genetics has an enormous range of education, clinical research, and healthcare. Human genetics also allows learning of innovations, keep the string of genetic knowledge alive, and pass on the knowledge gained to the next generation. Human genetics also helps in understanding and treating genetic diseases and genetically influenced ill health. Human genetics research also deals with the mechanisms of human gene function, malfunction, and treatments. Because of a high degree of evolutionary conservation between organisms, research on model organisms that are easier to study often provides valuable insights into the human genome function. In 1963, the WHO expert committee observed the role of genetics in the health and well-being of persons living now and also future generations. With the development of advanced technology, the World Health Organization program of hereditary diseases was started in 1980s and expanded its focus on the genetic approach to prevention and control of major hereditary single gene diseases and non-communicable diseases. Further, to facilitate global networks of experts to address multiple issues on human genetics, WHO has made provisions for on-site support for the delivery of genetic services to expand partnerships and collaborations, especially with low to middle income countries. The Human Genetics Program is also addressing various genetic issues of ethics, legal, social implications, and the global arena of public health. Now, the internet revolution has changed the face of communications in our world and is making global communications and networks faster and more far-reaching. This will help to understand the genetics and expand the possibilities for participation of public engagement, information exchange, and representation, especially for low to middle income countries. Finally, complex genetic diseases are the focus of a great deal of research that has carried out today. Therefore, the knowledge of human genetics helps to contribute translational research, which in turn to the healthcare industry in terms of a new effective diagnosis and treatment option for human diseases. Dear students, with this, we come to the end of this module on history of human genetics. See you with the next module. Till then, bye-bye, take care.